Hi, today I'd like to tell you about targeting the sigma-2 receptor for dry AMD, age-related macular degeneration, and why we think it could be a promising therapeutic approach. Age-related macular degeneration is a leading cause of blindness, particularly in those aged 65 and older. 11.2 million people are currently afflicted, and that's in the US alone. Patients suffer a loss of central vision due to deterioration of the macula. This causes a blurred central vision as shown here, and eventually this vision is lost, making daily activities early on difficult and later on impossible. There's two types of late-stage AMD. One is the dry form, late-stage geographic atrophy, and another is the wet form, which involves an exudative vasculature type of pathology. The wet AMD is more severe with respect to the loss of vision function, but it's less prevalent and treatments do exist. For dry AMD or geographic atrophy, this is the more prevalent form. 90% of all cases of AMD are dry, and there are no approved therapies at present. So I'd like to take you through the cellular and molecular pathogenesis of dry AMD in a little more detail. What's depicted in the schema on the left is cellular layers of the retina, which is the tissue relevant to AMD. And in particular, in this red box shows where the AMD pathogenesis begins. The Bruch's membrane, the RPE layer, and eventually the photoreceptors. And to depict the molecular events in more detail, that's shown here on the right. What you can see here is that in early dry AMD, drusen develops, which is an accumulation of lipid deposits as well as some proteins. And this causes a thickening of the Bruch's membrane and disrupts the cytoarchitecture of the retinal pigment epithelium. Subsequently and progressively, this impacts the health and functions of the RPE cells and the photoreceptors. This can be often initiated by just a lack of nutrients to the RPE, impaired transport to that region due to the drusen's presence, and also through oxidative stress and inflammation that accumulates here, leading to ultimately cell death of both RPE and photoreceptors. So several disease biologies that are relevant to dry AMD have been targeted through multiple approaches in the clinic, pursued by pharma and different biotechs. And unfortunately, there haven't yet been any successes that have led to a product on the market that patients can have access to. Many approaches also require invasive treatments, gene therapy, stem cell replacement, and intravitreal injections. That would be an injection into the eye. So non-invasive small molecule approach would be ideal. And ultimately, there is a dire need to develop and test novel therapeutics in the clinic. Evidence has accumulated over the past years that targeting sigma-2 receptors in dry AMD may be a promising therapeutic approach. Sigma-2 receptors are expressed in the target tissue, the retina, as well as the cell types relevant to AMD in the retina, notably the retinal pigment epithelial cells and the photoreceptors. Secondly, human genetics points to a role of TMEM97 in dry AMD, and I'll take you through the details of that on the next slide. In vitro proof of concept studies indicate a protective effect of TMEM97 knockdown in a cell-based model of AMD. And there's also a strong biological link between functions of sigma-2 receptors and the disease biologies relevant to dry AMD. Finally, an opportunistic study that we've done, we leveraged our clinical trial proteomics data from aged, in our case, Alzheimer's disease patients treated with the sigma-2 receptor antagonist CT1812 or placebo. And this data that I'll show you provides early proof of principle in human patients that sigma-2 receptor antagonists have the potential to alter proteins and pathways in dry AMD. So 2016 early genome-wide association study, the GWAS study, identified a SNP in the TMEM-VTM locus that alters the risk of dry AMD. And this provided the first human genetic evidence implicating TMEM97 in dry AMD. So that's really exciting. And since then, it's notable that independent studies have identified the same locus, and these have been large-scale GWAS studies. This SNP here, of course, is the minor allele, actually confers a decreased risk for AMD compared to the major allele. And further study will be needed to determine if and how the single nucleotide polymorphism affects TMEM97 expression or function. But this is really exciting. One group, the Wang et al. Group in 2020, looked at this more carefully. Based off that genetic evidence, they wanted to see what TMEM97 knockdown would do if you knocked it down in a cell model of AMD. And so to do that, they used the ARPE19 cell line. It's an RPE cell line. And they exposed the cell line to oxidative stressor to elicit cell death. And what they did first was they had cells that had normal constitutive levels of TMEM97, and those that were knocked down, so had reduced levels of TMEM97 using CRISPR technology. What you can see here is plotted here is cell viability. And you can see here that when these cells are exposed to oxidative stress, in this case, the stressor tert-butyl hydroperoxide, there's a drastic decrease in cell viability. In cells that have knockdown of TMEM97, but no oxidative stressor, this does not alter cell viability. However, when presented with the oxidative stressor, the amount of cell death that is elicited is significantly reduced compared to the same cells that express normal levels of TMEM97. So this is really exciting. Whether or not pharmacological perturbation of sigma-2 receptor signaling mirrors this genetic knockdown of TMEM97 is currently underway, in addition to evaluating additional mechanisms by which sigma-2 antagonists may be efficacious. Based off known roles of sigma-2 receptors, we hypothesized that sigma-2 antagonists may be beneficial in ameliorating several pathways disrupted in AMD. And that's what's depicted here on this slide. As this audience is fully aware, we know that all of these biologies are, or can be, altered by sigma-2 agonists and antagonists. And I've also highlighted here this slide that depicts some of the key biologies that are involved in AMD. Those in the red boxes are biologies that are known to be affected by sigma-2 in other systems, or in the case of oxidative stress in RPEs themselves. So targeting sigma-2 receptors may enable a promising multi-mechanism therapeutic approach. We performed an opportunistic analysis of our Alzheimer's disease clinical trial proteomic data, and to try to understand whether or not it was possible to move proteins relevant or in pathways relevant to AMD. So I'll briefly describe the study design and the high-level takeaways. So what we've done is we took our recent SHINE-A proteomics data. And of course, this data was initially queried for the purpose of understanding how CT1812 affects the proteome in Alzheimer's disease patients. And so for the SHINE-A clinical trial, these are mild to moderate Alzheimer's disease patients that were given CT1812 for 6 months, or they were given placebo. And so from this, we have a list that we generated of proteins that are differentially expressed in CT1812 versus placebo in this patient population in CSF and plasma. We simply asked, what is the overlap of those proteins that are differentially expressed by CT1812 with proteins that are known to be disrupted in AMD, either in tissue such as derived from AMD in control patients, or from plasma derived from AMD in control patients. And so these are the data sources listed here from which we procured these proteins. We identified a set of proteins that indeed overlapped. So that means these proteins were altered by CT1812 that were known to be disrupted in AMD. We then subjected them to string protein network analysis. And you can see here, this is a network of proteins that are highly significantly connected. And from this, we asked what were the top most significant biological processes. And what you'll see in this list, and all of these are significant, as you can see here, you'll see that many of these were shown in the previous slide of known AMD-related biologies, including complement, proteolysis, extracellular matrix organization, inflammation, and response to stress, as well as positive regulation of transport. So this study, again, it was opportunistic, but it really did enable the identification of a subset of proteins preliminarily. and pathways genetically linked to or known to be dysregulated in dry AMD that were regulated in 80 patient biofluids, given those 80 patients that were given CT1812 versus placebo. This provides preliminary proof of principle as well, that the sigma-2 antagonist CT1812 may be capable of altering AMD-related proteins and pathways in an aged, in this case, Alzheimer's disease population. So this was really exciting. And in terms of moving forward towards progressing a sigma-2 antagonist for AMD, we didn't know in these Alzheimer's disease patients whether or not CT1812 was reaching the eye tissue, the retina tissue, and what the drug exposure levels were. So we instead did a study in rat. So what we did was we took C14 labeled CT1812, gave that via oral administration to the rat, and then did a pharmacokinetic study where we simply asked what were the drug levels in both brain and in the retina tissue, shown in orange, over time. And so notably, as you can see here, the exposure levels in the retina were deemed to be quite similar to that in brain. And this is really exciting. It provides confidence that through an oral administration of CT1812, we may be reaching the target of interest for dry AMD. And this positions us to be able to test the hypothesis that sigma-2 antagonism with CT1812, that it will be efficacious in dry AMD. Also want to point out that these drug exposure levels have been modeled for Alzheimer's disease based off in vivo preclinical efficacy studies. And as you can see here, our goal is to achieve greater than 80% receptor occupancy to achieve efficacy for Alzheimer's disease. And you can see we are well above that line with the exposure levels in the retina. So we would expect this to be a therapeutic dose. So the take-home messages and future directions that I'd like to leave you with is that targeting sigma-2 receptors for dry AMD is a promising therapeutic approach. Being able to target sigma-2 with a small molecule approach is advantageous, and that if given orally, if this mechanism of action is efficacious, this would enable a non-invasive therapy to be provided to patients with dry AMD. We have plans to advance CT1812 to the clinic for dry AMD with an oral administration this year. And preclinical proof of concept studies in progress will further elucidate the mechanisms by which sigma-2 antagonists, including CT1812, will inform upon the appropriate patient selection, time to intervene, and the clinical outcome measures to enable a successful clinical trial design. I would just like to acknowledge the team at Cognition, our board of directors, our advisory board members, both Alzheimer's disease as well as our dry AMD advisory board members, as well as our industry collaborators, including Proteome Science and Ian Pike, who provided us with the proteomics measurements for the clinical trial work, and our AMD academic collaborators. Thank you.